Welcome to Lecture 5 for Medical Physics 671, Human Physiology and Anatomy. Well, let's continue our journey on the physiology of excitable cells. We'll cover muscle energetics, fatigue, and training. The textbook has this figure shown in the left side of this screen that shows the excitation of different muscles within the rat leg as the rat is walking. And it's not really central to the other information in this chapter. It's just a way of showing how the muscles coordinate with each other. And I thought it was an interesting example. So the excitation of the muscle that occurs first is with this shin bone muscle shown down here in this rat diagram. And that makes sense. You got to pick up your foot, so that muscle has to contract in order to move the foot up. And that happens very quickly and very sharply. Then there is a muscle in the back of the leg as well, and that helps to bend the knee. So this muscle shortens in the calf of the rat, and that can be a longer excitation period to move the leg forward and bend that knee. And then the foot drops, that excitation stops. And then two muscles take over from there. The thigh muscle on the outside, and there's an inner thigh muscle as well. And you can see that the excitation happens for an even longer period for those muscles, occurring with the bending of the knee and moving the leg forward as well. You also notice that there's an excitation of the muscle in the thigh right after lifting up the foot. And I think that's to stabilize the leg as it moves forward and drops back on the ground. So it's interesting to see how these muscles are coordinated with each other. All right, well, let's get back to the main part of this chapter. The book describes that muscular activity consumes a lot of ATP at high rates. Well, I think that's pretty obvious. The major consumption of ATP occurs when you have that myosin head group binding to actin. So there is a lot of ATP that is consumed in that process. But there is ATP consumed at other parts of the process as well. There are active transporters that move the sodium ions back to the outside of the cell against their gradient. Calcium transporters that move calcium outside of the muscle cell against their gradient. Now you have active transporters of other ions as well. But the large majority is taken up by the myosin-actin cross-bridge cycling. So there are three ways to regenerate ATP. The first is shown here, where phosphocreatine donates a phosphate group to ADP to create ATP, and as a consequence, phosphocreatine is converted to creatine. Now this can happen very rapidly, as you can see in the table down below. Creatine phosphate energy source can be consumed to make new ATP very quickly. But the amount of ATP that can be created is actually quite low. And this is really to maintain a buffer of ATP near the myosin head group. It doesn't supply a lot of new ATP to the muscle. Instead, the glycolysis pathway and the oxidative phosphorylation pathway, shown in this diagram on the right, is really where you get all of your new ATP. So glucose enters the cell through a glucose transporter. GLUT1, GLUT4 are pretty common. And the initial step of metabolism is to convert glucose to 2 pyruvate molecules from 1 glucose molecule. And this is called glycolysis. This first consumes 2 ATP molecules, but then produces 4 ATP molecules, so you get a net of 2 ATP molecules. Now, if you continue with glycolysis, or sometimes known as anaerobic glycolysis, because it doesn't require oxygen, the pyruvates are converted to lactic acid, and the lactic acid is expelled from the cell. Also, you'll notice that NAD plus is converted to NADH during glycolysis, and that gets converted back to NAD plus during anaerobic glycolysis. So there's a second reason for this extra glycolysis step, is to replenish NAD plus. That's needed in the first steps of glucose metabolism. Or you can go through oxidative phosphorylation. The pyruvates can enter the mitochondria, the inner part of the mitochondria, not just in this intermediate zone between the outer membrane and inner membrane of the mitochondrion. And, technically, you'll see textbooks list that there are 38 ATP molecules produced by the whole process through oxidative phosphorylation, or perhaps only 36 if you ignore the 2 ATP molecules that are made in this first step up here. However, in practice, you only get a net of 30 to 32 ATP molecules when you really do measure muscle physiology. And the reason for that, which we won't go into detail here, is that inside the mitochondria, the pyruvate is converted into FADPH and NADH. And then that is used to eventually drive an ATPase pump that creates ATP due to a proton gradient. Well, all those players in between, FADH2 and NADH, and also the proton gradient, well, they can leak a little bit. They're used for other purposes as well. And so you don't get the full benefit of 36 or a total of 38 ATPs out of this process. Still, 30 to 32 ATPs is a really good haul for a glucose molecule. Glycogen to lactate glycolysis, and then I'll call it anaerobic glycolysis. That happens somewhat rapidly, and you get some ATP out of this process, but clearly you get a lot more ATP if you go through oxidative phosphorylation, which happens much more slowly, many more steps involved. Also, fatty acids can be converted to ATP through other metabolism, not shown here. You get a relatively slow rate of ATP production, but you get a huge amount of ATP. And there's a show called Alone, where people go out into the wilderness in Alaska and try to live alone. And the winner is the one who can live alone in the winter in Alaska with minimal supplies. And that's kind of a cool show. Well, one of the best survivors in the show Alone actually was able to get enough fat in his diet. He killed a moose and was very compassionate about that, but was able to survive off of the moose. And the others were not. And you can see how much ATP you get from fat. And it's really tough to live on rabbits out there in the wilderness because they just don't have much fat. And in fact, our ancestors years ago had a fat-based diet. They weren't growing crops and making Fruit Loops and Cheetos to eat. So they had to survive on fatty acids. This lactic acid production actually is more interesting than just that previous slide. First, I guess I should mention that we talk about a anaerobic threshold. Your muscles have a lot of oxygen, and then when you can't keep up with the oxygen supplied to the muscle, well, the metabolism is may switch over to the anaerobic glycolysis rather than the oxidative phosphorylation mechanism that produces a lot of ATP. Then you have less ATP, and you have more lactic acid buildup, and we know that we feel the burn in our muscles. We feel that muscle fatigue when we're running low on energy. Well, that's the old school thought that there was some sort of light switch on-off mechanism to go from oxidative phosphorylation to anaerobic glycolysis and the production of lactic acid. But I think we have a more sophisticated understanding of how this works now, and both mechanisms are always present. It's just that after a while, when it's more difficult to get oxygen to muscles, or you're running low of ATP and you got to make it quickly, well, there's a more of a balance of anaerobic glycolysis and the production of lactic acid and ATP, and a little bit less oxidative phosphorylation going on. But both always exist all the time. So the anaerobic glycolysis increases, but it isn't a switch where oxphos is turned off completely. The second point here is that lactic acid is shuttled into the inner mitochondria, as shown in this blue diagram, and the lactic acid is converted to pyruvate. And then pyruvate can go in the TCA cycle, this oxidative phosphorylation cycle, and make a lot of ATP. And there is some energy penalty in converting glucose to pyruvate to lactic acid, and then having lactic acid come into the mitochondria. But it still is a way of recovering a fair amount of energy from the lactate, which otherwise would just be a waste byproduct of the anaerobic glycolysis. Or as a third mechanism, lactic acid is shuttled to other glycolytic fibers and converted to pyruvate in their mitochondria. So you can feed other muscle cells with the waste byproduct, lactic acid, of anaerobic glycolysis from one muscle cell into another muscle cell and create energy for those adjacent muscle cells. So that explains why you have lactic acid build up in your muscles and in that extracellular space as well, because there's transport of lactic acid between cells. And then even more interestingly, lactic acid is shuttled to the liver and is converted back into glucose, or even more so glycogen. Glycogen is actually a very large source of energy, a polymer of glucose monomers, and that can be used by muscles as well, eventually broken down to glucose 6-phosphate and enters the whole metabolism process. That would explain why lactic acid buildup is seen in your blood, and the blood can become slightly acidic. If you're interested in a cute movie, you can go to this link here and watch PepCK Super Mouse, a genetically modified mouse that had upregulation of the PepCK enzyme, which greatly increases or facilitates this process of converting lactate to glycogen. And the movie shows that the mouse with this mutation has an enormous amount of energy and can run for hours relative to a normal mouse. Can run 3 miles. Can anybody run for 4 hours? Can you run at a sprint for 3 miles? That's pretty incredible for this little mouse. I encourage you to look at that video. Okay, well, the book goes on into a lot of extra detail about muscle physiology. I'm not going to show any figures from this part of the chapter. Instead, I just want to emphasize some points that the book makes. First, exercise increases glucose transporters in the muscle sarcolemma. That would be the wrapper around the muscle filament. Insulin will stimulate the recruitment of those transporters, the GLUT4 transporters in particular, from vesicles that are in the muscles to get them to the sarcolemma so that you facilitate the absorption of glucose from the bloodstream into the muscle. And patients who have diabetic condition will have difficulty getting glucose into their muscle cells, and so there's a lot of glucose in their bloodstream. Now, contractile activity will also stimulate the recruitment of GLUT4 from these vesicles and improve glucose transport into the muscle. So diabetic patients have to be a little careful that they have to reduce their insulin injections, which help to boost the amount of glucose coming into the cell, because they're naturally boosting the amount of glucose coming into their muscle cells due to contractile activity and the exercise that they're doing. With proper monitoring of the amount of glucose in the blood system, diabetic patients will be okay. So, not surprisingly, during high-intensity, short-duration exercise, fatigue occurs. It's interesting that that's caused mostly by an increase in the cytosolic inorganic phosphate. So if you think about it, myosin is binding to actin. That's released when ATP binds to the myosin head group. The ATP gets hydrolyzed into ADP and inorganic phosphate. The inorganic phosphate leaves the head group, and that allows the head group to move in its orientation. Well, you build up a lot of inorganic phosphate then when the phosphate is released from the myosin. If you have a large amount of inorganic phosphate next to that myosin that is bound to phosphate and ADP, well, then the inorganic phosphate on the myosin is less likely to leave. It's going against its gradient to that higher concentration in the cytosol. So short duration exercise builds up a lot of inorganic phosphate that cannot be washed out of the system or converted back to ATP. And so that is a good mechanism to reach fatigue. Tell your body, look, you got to wait and let me catch up on my metabolism here in the muscle. During lower intensity, low duration exercise, fatigue is caused by glycogen depletion. So once you run out of the fuel during a low intensity, long duration exercise, well, the muscles just don't have enough fuel, don't have enough glucose to turn into ATP and run out of energy. All right, winter's coming up. I'll eventually head over to the Baki Rec Center, sign up again, and I'll be the old guy on the treadmill on the 4th floor. But when I first do that, my muscles are going to be sore, and I'm not really building any new muscles. Instead, what I'm really doing during the first 2 weeks of getting back in shape on the Stairmaster is to do initial improvement of how my nerves are firing from my brain into those muscles, the activation of nerves. Then further training will increase the diameter of the myofibrils in the muscle So those will get larger. You'll have more myosin and actin filaments or polymers within the myofibril. But you don't recruit more myofibrils. Instead, it's really longer-term training that increases those number of myofibrils and the innervation of those T-tubules and the sarcolemma to innervate, and also the glucose transporter mechanisms become more abundant. And so you just build muscle, right? Well, that takes long-term training. And unfortunately, when people go out to exercise, they're hoping to see that buildup of new muscle mass within a relatively short time. And unfortunately, it takes, on average, a week or two to just get your nerves firing in a more coordinated way to work on your muscles. And then also you do get some muscle growth intermediate, but it's really the long-term training that increases muscle size, as we all can surmise. All right, so endurance training really doesn't improve the muscle mass. Instead, it improves the efficiency of muscle metabolism without increasing the muscle size. Okay, so let's go on to smooth muscle, the last section of this chapter 3. So smooth muscle is different than the striated muscle, the skeletal muscle or the cardiac muscle that we've been talking about. It doesn't have as much uniformity or unidirectionality to it. And therefore, if you observe the smooth muscle or look at it under a microscope, it looks more smooth. Well, smooth muscles line hollow organs, such as the intestines, or tubes, such as the arteries. And that controls the diameter of the arteries. You can get vasodilation or vasoconstriction to change blood flow and blood pressure. You can change the diameter of the small intestine in a coordinated way to push food through the intestine. Okay, so these smooth muscle cells are about 2 to 5 microns in diameter and 400 microns long. The skeletal muscles, the striated muscles, tend to be much longer and multinucleated. These smooth muscle cells are only contain one nucleus because they're relatively small. And then most importantly, smooth muscle cells are controlled by nerves, just like the striated cells. But they're also controlled by hormones. And so you do not have to have electrical activity from your brain to a smooth muscle cell in order to get the smooth muscle cell to contract or relax. You can have chemicals cause muscles to contract or relax. Now, the book describes that there's a lot of temporal activation of smooth muscle cells. It can be periodic, it could be sustained, it could build up over time, and so on. And I really won't dwell on that in this lecture. Also, you can look at the amount of calcium that is stimulating the muscles. Recall that calcium is an intermediate that turns electrical signals into muscle contraction. You can see that there are different increases in calcium that would also allow the muscle to be activated. You could have calcium be high for a long time and activate the contraction of the muscle for an extremely long period of time. All right, well, this is the main diagram that you really have to pay attention to. And I know it looks complicated, but we're going to go through it step by step. First, we have to get calcium influx into the muscle. As you may recall, the calcium is responsible for binding to troponin C. That then interacts with tropomyosin, a protein that hides the binding sites on actin so that myosin cannot bind to actin. We need calcium influx to remove that masking effect. And there are 3 ways of doing that. First, you can have voltage-gated calcium channels just with nerve stimulation like we've been talking about. You also have a storage of calcium outside the cell. Naturally, calcium is more concentrated outside of cells than inside of cells. And so you can have a membrane transporter protein system open up and allow calcium to naturally diffuse from outside the cell into the cell. The sarcoplasmic reticulum, this large structure inside the cell, near the cell outer membrane, and sometimes I'll just call that the SR, the sarcoplasmic reticulum. Well, that's where the calcium is stored inside the cell. You can have calcium ready to go when you need to contract the muscle. Well, a protein called STIM1 can recognize how much calcium is inside the cell. And when there's less calcium inside the cell, then it loses its calcium that's bound. And that triggers a conformational change in the protein, which changes these linkages, which influences the shape of the transporter and allows calcium into the cell. And then finally, you can have receptor-operated channels where a hormone or a small molecule can bind to the protein and open it up and allow calcium in as well. So you can have voltage-gated or molecular-gated channels. Okay, the ryanodine receptor, not super interesting. It's just a membrane transport protein that allows calcium to diffuse out of the highly concentrated calcium depository within the SR into the cytosol and then go wander over to troponin C and do its work. So I think a more interesting system is inositol triphosphate, which binds to IP3 receptors, particularly in the deeper part of the sarcoplasmic reticulum that's deeper in the cell, more so than on the superficial part of the SR that's near the plasma membrane, the outer membrane of the cell. It's key to take a look at this side over here and see how it interacts with IP3. An agonist is something that causes activity, will bind to a G-coupled protein receptor on the membrane of the muscle cell. And that will cause an activation of phospholipase C, which then produces IP3. So you get a lot of buildup of IP3 triggered by this hormone that binds to the outside of the muscle cell. And then the IP3 can bind to the IP3 receptor and allow calcium outside the cell. How does calcium then leave the cytoplasm, and how does the muscle then relax as the actin filament is then masked by tropomyosin again? Well, SERCA2 is a membrane transporter system that is modulated by phospholamban, and that allows calcium back into the SR, or you can pump calcium against its gradient from inside the cell to the high concentration outside the cell using active transporters that use ATP. There's a sodium-calcium exchanger, and then a protein called PMCA that's used. Okay, so if we move on to the smooth muscle cell itself, what does it look like? Well, the myosin and actin filaments create these contractile filaments that are shown throughout this cell, and they're anchored to these dense bodies inside the cell and then also to one point of the membrane. These contractile filaments do not attach from one membrane to another. They connect to these pillars, the dense bodies. The smooth muscle cell also has caveoli, so there's a much larger surface area, and that facilitates interaction with the environment. Also, these cells are coupled to each other, and you can have flux of calcium from one cell to an adjacent cell so that these cells do work in a coordinated way. Keep in mind that there isn't a nerve activation of these cells. Nerves which might innervate multiple muscle cells, for one nerve innervating multiple muscle cells and allowing for coordination. Well, that's not occurring if there is a hormonal activation of the smooth muscle cells. So the smooth muscle cells talk to each other. And in fact, the release of these neurotransmitters is not focal and very directional like it is in striated muscle. Instead, it diffuses throughout the cell, and that helps for all the cells to coordinate with each other. Oh yeah, also, if there is a voltage activation of a smooth muscle cell, well, that electrical activation can also be transferred to nearby cells as well. So there is some coordination there. Okay, well, phosphorylation regulates muscle contraction, and this happens in striated muscles, but it's really a common topic to discuss for smooth muscles because it's a way of having a switch. Either you're locked in to being contracted, or you're locked out and you cannot contract and you have a relaxed muscle. And we don't really want that with skeletal muscles as much. We want our skeletal muscles to be actively contracting and relaxing, but the smooth muscles can contract, and then we want them locked in place. And this phosphorylation does that. Well, here we go again with our myosin molecule and the two head groups and the two other myosin light chain proteins. Well, if you then were to phosphorylate with a kinase enzyme, you can phosphorylate the regulatory light chain, one of the two light chain myosin proteins. Then you can undergo contraction by having the myosin head groups bind to actin and go through that whole cross-bridge cycle. And that's shown in this inner diagram right here. I know this is complicated. Just focus on the inner part here, and you'll see, oh yeah, look at that. I've got ATP binding to the myosin head group, and that releases from the actin filament, and then ATP gets hydrolyzed. the inorganic phosphate leaves, and the head group changes its orientation and is bound to the actin filament, and then finally it's released again when ATP binds, takes the place of ADP, and so on. But all of this happens because there is a phosphate group on that regulatory light chain. And all of this on the outside here does not have the phosphate group on that protein. So you can turn off the cross-bridge cyclization at various parts by dephosphorylating. I guess we're dephosphorylating down here by the myosin light chain phosphatase enzyme that removes that phosphate group. And so the sections on the outer part of this diagram are frozen. Now you cannot have muscle contraction or relaxation. The myosin and actin are frozen in place. So this is a reminder that these figures and tables are not covered in my lecture here. I would consider them optional material. Now there might be some text that relates to those figures that are interesting. So I wouldn't say you should skip them, but I just want to show that there's not as much emphasis on those figures and tables, and hopefully that will help you get through the reading faster. Okay, then finally, this is the anatomy homework for this lecture. We're interested in the skull. This is a real basic diagram of the main bones. If you're interested in the skull, you can go through the skull in more detail as well. And then also the nasal region is shown here, and it's good to have a feeling for the parts of the nose, the cartilage, and the upper jaw. So I will go through this diagram as well. It is required material. And once again, if you really want to study the nose in excruciating detail, here's a much more complicated diagram as an option. And that brings us to the end of Lecture 5. It was a pretty short lecture. I think in class we shared some extra side stories. We watched that video, which was fun to talk about. And then that concludes the musculoskeletal section of physiology and anatomy. And we'll move on to Lecture 6, which will be the nervous system. So time to get nervous. I'll see you for Lecture 6. Thanks for watching.